Hi, this is Diptikan Padiari, lecturer in botany. Welcome to my channel Mikan's Botany. Please subscribe my channel and hit the bell icon. Hi, this is the third part of prokaryotic DNA replication. What happens when polymerase 3 will loaded? What will happen when DNA polymerase 3 will get loaded? What will happen? As you know, DNA polymerase has the two beta clamp and two cores of unit and one gamma complex. What will happen if this is the DNA? Sorry, it will be the some. If this is the DNA, if this is the two strand of DNA, what will happen? It is the 3' direction. It is 5', 3'. RNA polymerase, RNA primers will get synthesized in 5' to 3' direction. Means 5' to 3' means in this direction, and in this strand will be in 5' to 3' direction. Okay. As we know, RNA polymerase can only add nucleotides in the 3' hydroxyl end. DNA polymerase DNA pol 3 can add nucleotides only at the 3' hydroxyl end. That's why. And so what will happen? Let's see. If this is the two strand of DNA, if this is the beta clamp loader, two cores of unit, and this is the beta clamp loader. Three prime, 5', 5', 3'. Here will be the 5' to 3' end, 5' to 3' end. The nucleotides will get attached in the 3' to 5' direction, like this manner. Okay, it will be like this manner. Okay, in this direction. The movement of this direction is the movement of RNA polymerase 3. This direction of DNA polymerase 3. Okay, let's see. If this is the primer, and here will be the primer. If this is the 3' to 5', 5' to 3'. This is the 5' to 3' direction. 5' to 3' direction. The nucleotides will get attached in this direction. In the 3' to 5' strand. In 3' to 5' strand, the nucleotides should get added in this direction, and in here will be in this direction. As the RNA polymers get moved in this direction, so new polynucleotides get attached in discontinuously in this strand, and but here will be another polymerase. If this RNA polymerase will be change its position to this this point, there will be attachment of new RNA primer like this. Okay, and there will be the addition of polynucleotides to it. Okay, but this strand synthesizes DNA discontinuously, but this strand will synthesize continuously, this strand will synthesize discontinuously. This continuous strand is known as leading strand, and this discontinuous strand is known as lagging strand. And these fragments are known as Okazaki fragments. Lagging strand synthesize discontinuously by short Okazaki fragments. Okazaki fragments, as it is discovered by the Reiji Okazaki, a scientist, molecular scientist, Reiji Okazaki. By his name, these fragments are known as Okazaki fragments. These Okazaki fragments are 1000 to 2000 base pair long. Okay, let's see. DNA polymerase 3 synthesize two strand. One is leading strand, and then that is lagging strand. Okay, leading strand in the direction of same direction of replication fork movement. But lagging strand synthesis will be opposite to the replication fork movement. Okay, this is the most important point. We have to remember these 2 points. In which direction will the DNA replication proceeds? Okay, after this, DNA polymerase 1 will get attached. The next enzyme will be DNA polymerase 1. It has the property to remove the primer, as this is the combination of both DNA and RNA strand. So this RNA primer should be removed from here. This action is done by the DNA polymerase 1. This removes RNA primer using the 5' to 3' exonuclease activity, and adding only DNTPs one by one. Here will be a question, why there is only DNTPs will get added, not RNTPs. This question upon you will get answer me, or if any doubt, ask me on my channel. Let's see. It is the main strand of DNA. the primer are 5' to 3'. Here is the 5' to 3' means, and the new nucleotides are added in this direction. Okay, these are the new nucleotides. There will be a gap. Here will be a gap. The DNTPs and RNTPs are not get attached here, so there will be a gap. This is termed as nick. This gap is known as nick. This is not a gap. This is a nick. After what will happen? These are the primers. These two are primer. These two get removed here. After this, nick translation will be done. 3' to 5'. Sorry, this primer is removed now. Nick translation. Nick translation is done by DNA polymerase 1. Okay. What is nick? Nick is the gap missing a phosphodiester bond. Nick is the missing of phosphodiester bond. Let's see. I will get in this direction. This is the previous strand, and this the new nucleotides get attached by removing the primer. But this gap is removed by the DNA ligase enzyme. The next enzyme will be DNA ligase. As its name suggests, it joins the two strands, joins this gap. Okay, to DNA ligase functions. To function the DNA ligase, there is requirement of energy. Source to create phosphodiester bond. Okay, these energies are NAD plus. As we know, the source of energy are NAD plus and ATP, nicotinamide adenine dinucleotide, and ATP, adenosine triphosphate. This is the source of energy in prokaryote. And this is the source of energy in eukaryotes. What will happen here? Let's see. First step will be the NAD plus get attached to the DNA ligase enzyme to form. Now I write the DNA ligase 2 enzyme and symbolized by E. AMP plus NMN, nicotinamide mononucleotide. How this happens? Let's see. NAD plus is the AMP plus NMN, and this AMP get attached to the enzyme, and NMN I'll remove here. This AMP is transferred at the lysine residues of the enzyme. This is the important point to remember, as it is from a question point of view, from which residue will it get transferred, the lysine residue. Next, what will happen after this? It will be the old strand. These are the new strand. This is the 5 prime. No, sorry, this is the 3 prime direction. This is the 5 prime direction. New strand. This is the old strand. AMP is transferred at 5 prime end of the nick. Let's see the structure, how it looks like. Old strand, 3 prime to 5 prime direction, 5 prime to 3 prime direction, and here will be the AMP. This is the AMP, and here will be the presence of hydroxyl group. Okay. This is the step 2. In step 3, the 3 prime hydroxyl will act as a nucleophile. AMP replaced and a phosphodiester bond is formed. In step 3, what will happen? The 3 prime hydroxyl will act as nucleophile. AMP is displaced and a phosphodiester bond is formed. Let's see. Means it acts as a nucleophile here, and AMP get removed, and here will be the phosphodiester bond. This is the old strand. This is the, here will be the phosphodiester bond. Okay. It is the DNA ligase function work. Then in the next session we will discuss about the termination of DNA replication in prokaryotes.